So this is the last part of PowerPoint that is going to be focusing on the movement of molecule across the plasma membrane. But in this case, we are focusing on the transport that requires some sort of energy input from the organism to allow the movement of these molecules. This process is known as active transport or active membrane transport. Now, there is two main breakdowns for active membrane transport. The first one is known as active transport, which is the scenario where your cells, you require a specific type of energy in the form of either ATP or concentration gradient to move the molecules across the plasma membrane against their concentration gradient, or vesicular transport, which we're only going to talk about two types of it, even though there are other types such as pinocytosis and phagocytosis, but we're going to only focus on exocytosis and endocytosis. Understand that any of these scenarios described on top require ATP for the process to carry it out and be crossed from the plasma membrane. The reason you use active membrane transports are either the solubles are too large for the channel, so they cannot pass through the channel, so you need to bring them in using either exocytosis or endocytosis, or remove them via exocytosis. Solutes is not lipid soluble, which means basically they're too big and they can't pass through, or they don't have the characteristic to pass through, as most of the phospholipid bilayers are carry lipid characteristic or have fatty acid characteristics, so it doesn't allow other molecules to pass through, or solutes is unable to move down the concentration gradient. So in any of these scenarios, and most specifically the active transport that is described on top, one of the key concepts that I need you guys to be comfortable with is the fact that these molecules, for the movement of these molecules, you need some sort of energy, and in this scenario, in the case of active transport, you move molecules against their concentration gradient, basically from low concentration toward high concentration, which is basically reverse of the process of diffusion. So let's focus on the first type, which is primary active transport. Primary active transport is the type of active transport that allows molecules to move against their concentration gradient by inputting ATP in the process. So the driving force, the keyword here in the active transport, or more specifically primary active transport, is the usage of ATP. So you hydrolyze ATP, you basically bring in water and remove one of the phosphates to move those molecules against their concentration gradient. This is the example of the primary active transport you need to know: sodium, potassium ATPase, or sodium potassium pump. In this scenario, as you can see, you have high concentration of sodium outside the cell, low concentration of sodium inside the cell. So going back to the scenario that I've shown you earlier, if you're looking at the concentration, sodium is low inside, high outside, and potassium, I don't know what happens, potassium is low inside and, high inside and low outside. So moving these molecules against their concentration gradient requires the input of energy and requires the use of sodium-potassium pump. So sodium-potassium pump is going to enable you to move 3 sodium out and 2 potassium in with the use of ATP. And you can see that ATP has broken down to ADP and phosphate to allow for these molecules to move. I need to highlight this, that sodium is moving out of the cell against its concentration gradient, and potassium is being pushed in against concentration gradient. 3 sodium out, 2 potassium in at every ATP that's being utilized. Secondary active transport is a utilization of energy in an indirect form to move molecules against their concentration gradient. Now in this scenario, which is a really cool scenario, is you actually have two molecules involved. One molecule is moving with its concentration gradient, and the other one is moving against its concentration gradient. So let's take a look at the first scenario we have here. Sorry, not this scenario. Let's take a look at secondary active transport. This is primary active transport. So in this scenario, you have high sodium outside the cell, low sodium inside the cell, and you have low glucose, the green hexagons, outside the cells, and high glucose inside the cell. Your goal is to move both of these molecules into the cell. But sodium is going to be moving with its concentration gradient, and glucose is going to move against its concentration gradient. So you are going to, or sorry, I should say your cell is going to use the energy of sodium moving with its concentration gradient to push glucose along with it to bring it in against its concentration gradient. Now, because both of these molecules are moving in the same direction, that is the definition of secondary active transport, specifically known as symporter. Transport of two different substances in the same direction. Now, if you're moving one molecule in, one component in while another out, so the arrows will be moving in the opposite direction, we use the word antiporter. Again, one will be moving with its concentration gradient while the other will be moving against its concentration gradient. The concept of antiporter and symporter is how those movements happen. If both are moving in or out of the cell, that's a symporter. And if one is moving in while the other one's moving out, that would be an antiporter. When we discuss vesicular transport, which is the second type of active transport, we usually require ATP to move that. Again, these molecules are typically relatively large, insoluble, or they have no lipid, they're not lipophilic, which means they will not be able to pass through their plasma membrane. So the two mechanisms we use to move these molecules are endocytosis and exocytosis. Endo means inside, exo means outside. Cyte means cell, so endocytosis means moving a component or vesicular transport into the cell, endo, inside the cell, and exocytosis means vesicular transport out of the cell. Now, as I mentioned, when we talk about endocytosis, we have a specific type of endocytosis called phagocytosis, which is associated with your white blood cells bringing in microbes or components that needs to be destroyed. They basically bring them in the using mechanism of endocytosis and destroy the bacteria or the infective agent. So let's take a look at what these pictures describing. The top picture describes the process of endocytosis. So let's say you want to bring this particle in. Notice that the plasma membrane is coming in and surrounding the component that needs to be ingested, in a sense, or carry the process of endocytosis. The plasma membrane wraps around it, and by the time the plasma membrane has surrounded it, you have created something called a vesicle. Now, this vesicle can be combined with your digestive enzyme called from your lysosome, and basically you break the components apart. Exocytosis, on the other hand, is you already have a vesicle inside the cell. That vesicle comes in toward the plasma membrane, joins with the plasma membrane, and releases its component to the outside space or outer extracellular space. One thing that is extremely essential for you guys to understand is the wall of the vesicle in either cases is plasma membrane. It is a phospholipid bilayer. It is identical in characteristic, which means you basically can bring in a component by creating a vesicle by wrapping a plasma membrane around it, or you can have that plasma membrane of the vesicle join the cell plasma membrane to allow it to release the molecules to the outside space. And that is referred to as, again, as exocytosis. The picture you see